Hello, and welcome to this Flowers Canada Growers webinar entitled Are We There Yet? The Road to Parvispinus IPM, presented by the Vineland Research and Innovation Centre, the Ontario Ministry of Agriculture, Food and Agribusiness, and also by Agriculture and Agri-Food Canada. My name is Margaret Beacock, and I am the Administrative and Project Coordinator here at FCG. All right, so today I am joined by Ashley Summerfield and Taro Saito, both of whom are researchers at Vineland, and Rose Labbe, a research scientist at AAFC, and Sarah Jandricic and Cara McCreary, who are both greenhouse IPM specialists at OMAFA in floriculture and vegetables, respectively. And I'm also joined by my colleague, Carrie Gates, who is the Pest Management Director at FCG, and I see we have Rose Boudenhouse as well from Vineland. So throughout the webinar, you can type your questions into the Q&A area so that they can be answered after the presentation. And I want to honor your time as you joined us on your lunch hour, so I will hand it over to Sarah.
Okay, I'm gonna share my screen, get it all set up. Okay, and if everyone else can just turn off their cameras until it's their time to go. Rose, you can stay up since you're almost next. Okay, everyone, hi, and welcome to this third and final webinar in our series on Thrips parvispinus control. So I feel like this has been a long road. Parvi first happened on the scene in 2021. It was a bit of a scramble at that time, so I'm sure at that time, if people could have hit Parvi with their car, they would have. We've done a lot of on-farm trials with pesticides and things, but now this is a time where we've started researching biocontrol solutions. So, in terms of what we're going to talk about today for this third and final webinar, Rose Labbe is shortly going to talk about thrips genetics and how Western flower thrips, our old thrip species, is different than Parvi in a lot of ways. So that's sort of tying back to our second webinar, contrasting Western flower thrips with Parvi. And then we're going to get into the meat and potatoes of the biocontrol part, which is looking at promising natural enemies, microbial controls and what role they play. And then we're going to talk about how you would integrate those into a IPM program for Parvi in both flowers and veggies, taking into account other potential pests. And you may notice that in this webinar and the past webinar, we focused a lot on using mandevilla as a host plant. And the reason for that is that it's really our worst case scenario crop. It has a really low tolerance to Parvi compared to the other floral crops out there and the vegetable crops. It's got tons of challenges. It's propagated at high temperatures and then grown at low temperatures. It's like one big crop per year, so you only get one kick at the can to get it right. It's got huge pot sizes with dense canopies. So basically, if we can get it right in mandevilla, we have a better chance of getting it right in other crops and taking what we learned from mandevilla and then dialing it back for less susceptible floriculture crops, things like anthuriums or hibiscus or veggies, and then hopefully making those programs less expensive as well. So with that, I will hand it over to Rose to talk about another way that Parvi is fun and different from Western flower thrips.
All right, thanks, Sarah. Can you hear me okay?
Yeah, you sound good.
Okay, sounds great. All right, I'm just going to try to flip slides. Okay, so hi everybody, and thank you so much for joining us. I am really excited to show the start of the work that this work is principally done by Avery Johnson, who's a master's student in my lab, and also in co-supervision by Dr. Rebecca Hall at the University of Guelph. Just to start us off in terms of the genetic diversity of thrips in Ontario greenhouses, let me see if the cursor works here. Oh, here we are. Okay, a little, too far, sorry, a little ambitious. There's a little lag. The thrips, here's a history of the thrips that we typically have in Ontario greenhouses. So back in before the 1980s, we had only one thrip species everybody was worried about. It was onion thrips. So this was a species that you'd find on all kinds of Canadian vegetable and floriculture crops prior to the 1980s. But in 1980 came along Western flower thrips, and it really started off a huge cascade decade from there on. And since then, this pest has been the dominant thrips species that we found in Ontario greenhouses and across Canada as well. Back in 2012, people have noticed in greenhouses that there were a growing number of unmanaged thrips outbreaks in Ontario. And the question was, what species are represented? And by 2016 and 2018, there were more systematic surveys of what thrips species were actually occurring, and there were some surprising findings. So there was actually a numerous species that were identified other than Western flower thrips. And of course, this work continued, especially with the surveys done by VRIC, and which were published in 2023, which found the first report of Thrips parvispinus in Canadian greenhouses. So in today's talk, I'm going to be talking a little bit about what thrips species we have, but I'm also talking about the genetics. And so it might not just focus on Thrips parvispinus, but I think it gives a little bit of a perspective for what diversity we have in Canadian greenhouses. So the questions we wanted to answer are: what thrips species are present in our greenhouses? Where do the new thrips species come from? And how do we approach management of an increasing diversity of these thrips species altogether? Moving on to the next slide. So just an overview of the 2023 survey. And you could see, as I have already described, on most of the crops that were surveyed to date, the great proportion of thrips species that were identified were Western flower thrips, followed by, of course, this kind of surprising, it was sometimes forgot about onion thrips, but certainly it was well represented by all of the major crops to some degree, and especially on cannabis, which had the greatest proportion of onion thrips, and of course the small number of other species that were part of that survey. But the question is, that survey didn't really look at greenhouse strawberry, which is of course an up-and-coming greenhouse crop in Canadian greenhouses. And then of course there's always the risk that we're going to find these odd other thrips species such as parvispinus, the chili thrips, and the tobacco thrips, which are always concerns for veg crops as well as ornamental crops. So things like this need to be identified early to be able to manage them effectively. And so we wanted to take a genetic approach to see if we can actually clarify what species we're actually finding through morphological ID and wanting to see if we can get confirmation. Why do we want to study genetic diversity of thrips? Well, there's so many benefits beyond just species ID. These can help us confirm the accurate species identification we're getting through morphological identification. It can help us devise and determine if we have species complexes and biotypes. It can track movement of sources and invasion pathways. It can help us detect different strains that are resistant to different pesticides. So in terms of insecticide resistant management, this is critical. It can also help improve monitoring and surveillance systems for early detection, and helps us understand relationships of specific strains to different host plant types. And of course, ultimately helps us pair and develop the appropriate biocontrol strategy for the different thrips species, because not all biocontrol programs work for all thrips species. And finally, it also helps us link thrips to specific plant viruses and vectors. And this is something that could help us predict what are the major threats for different crops. And there's just an overview of the different thrips species that we have in Canada, and their potential range of vectors, viruses that they vector. So this is obviously just an illustration, but the genetic barcoding could help us to devise a list that is very accurate for growers in Canada one day. Now, moving on to how do we assemble the genetic information? Well, first of all, we start with the genetic barcoding, which is based on the mitochondrial cytochrome oxidase gene. And based on that information, we can assemble these haplotype network analyses that tells us the relationship between the different thrips species. So the size of the circle represents the number of individuals that share one haplotype, which basically means one genetic variation. And the number of dashes between the lines of each of the circles, linking each circle, tells us how much genetic variation there is. So if there's 3 lines, there's 3 nucleotide differences. So this tells us something about the relationship between the haplotypes. So for the survey, this work was done by Avery Johnson in collaboration with folks at Vineland. So folks at Vineland helped us collect yellow sticky cards in the Niagara region. So there were 3 farms involved there. And there were a total of 6 farms involved in the Essex County and Kent County regions. And here's a bit of a schedule for the collection of yellow sticky cards from which the thrips were monitored. And you can see there's a little bit of variation depending on the crop type or the seasonal cycle for the different crops. But it basically covered the period from August 2025 all the way to May 2026. So just a basic overview of the results that Avery alluded to, basically through first morphological identification of the thrip species on yellow sticky cards. You can see, first of all, the abundance is quite variable, probably affected by crop type or the seasonal abundance of thrips, depending on where the crop is in the seasonal cycle. And obviously, there's a little bit of variation in terms of species identification. And of course, a little bit more detail can be seen in the next figure here. You can see, obviously, for all of the four crop types, 3 vegetables and 1 ornamental crop, mums being the ornamental, we can see that the dominant species is Western flower thrips, followed by soybean thrips, and actually an onion thrips is a big representative on some ornamental crop types, but it's certainly present on tomato crops in the Leamington region as well. For strawberry, we found mostly Western flower thrips. Now, in terms of the identities and confirming these through genetic barcoding, do they actually match the morphological identifications? Well, it turns out that in most of the cases, this is the answer is yes. But in the case of one site, a tomato greenhouse, we actually got the wrong identification through morphological ID. So if we dig a little bit deeper in that particular site, we see that of the specimens submitted for DNA barcoding, instead of having 4 Western flower thrips represented in that site, they actually turned out to be 4 Eastern flower thrips. So a completely different species of Frankliniella. So this is Frankliniella tritici instead of occidentalis. So what's interesting about this is that they're almost completely morphologically identical, and they actually fell into the same morphological class based on the use of the key. But if we look a little bit more closely to the morphological features that distinguish those 2 species, you can see, I think this is a really useful particular trait to be looking at, is the pronodal setae, which we obviously use for many distinguishing between many other thrip species as well. But you can just see that in the case of the Western flower thrips, there's a total of 4 median setae, whereas in the case of the Eastern flower thrips, or Frankliniella tritici on the right, there's only 2 median setae. So this is could be one helpful character to help growers distinguish between them. Now, we obviously didn't really find Eastern flower thrips previously in the greenhouses, but we certainly did find it in the fields, and this is a result of the field survey for thrips in different counties around southern Ontario. And you can see, obviously, Essex and Chatham-Kent have primarily Western flower thrips, but in the case of Middlesex, Elgin, Norfolk, Niagara, and Durham, actually a large proportion of field crops in those regions were represented as having Eastern flower thrips, which are obviously the major sources for these thrip species. Something to keep in mind. Now, what about the genetic diversity if we actually look at the haplotypes of these different thrips that we're barcoding in this study to date? And now this is just a very beginning of the analysis, but for onion thrips, we only found a total of 2 haplotypes who are very low genetic diversity. It makes sense because these are parthenogenetic insects mostly. With respect to Eastern flower thrips, we found only a total of 3 genetic haplotypes. So again, very low degree of genetic variation. In the case of Western flower thrips, now this is where it gets interesting. There were a total of 11 distinct haplotypes identified from 4 sites and from 3 crops. So this is, you can see the representation of thrips from Niagara, Leamington, different sites and different crops. Very complex relationship between these thrip species, and very interestingly, there's one site which has a very unique lineage of Western flower thrips, which is represented by 18 different changes, different mutations in that particular barcode region. Well, what does this mean for us? It actually means that there's probably a cryptic species of Western flower thrips that exist in Ontario greenhouses that will probably respond differently in some senses in terms of the management approach that we take on different crops. So something we need to be aware of the prevalence of. Another study also showed that there were cryptic species of Western flower thrips. A study by Rugman and Jones from the California populations of Western flower thrips showed, again, a very similar distinct lineage. 17 changes to the barcodes were found here. This suggests there's reproductive isolation, which leads to the development of a cryptic species of Western flower thrips that are, despite the fact that they're morphologically identical, there might be important implications in terms of the differences in their biology, their behavior, or even resistance to pesticides. So something we need to look at more carefully, and we're just starting to understand. So moving along, Thrips parvispinus, I'm almost finished here, but just to say that with Thrips parvispinus, not surprisingly, had very little genetic diversity in their population, so that's good. Overall, the greatest diversity that we found in the study was Western flower thrips. And the second important conclusion for the study is that Eastern flower thrips can be distinguished effectively through DNA barcoding. But certainly now that we're aware of the occurrence, there are morphological traits that we can hone in on to distinguish those species from the other Western flower thrips. Overall, I think it's important to realize that different thrip species have different management implications. Onion, soybean, and Eastern flower thrips tend to be fly-ins, so they're managed slightly differently. And you can use, obviously, tools like mass trapping for mitigating their population. In terms of Western flower thrips, however, things like cleaning between crop cycles and dipping for plant material is going to be important because these tend to be established populations that come in from plant material that are carried on from other crops from last year, for instance, or from other locations. So something to be considering when managing different thrip species. With this, obviously, for future research, we want to look to answer questions like how genetically diverse are the thrip species in Canadian greenhouses? We've just scratched the surface. We just started to do the barcoding, but we have much more to discover in terms of diversity throughout Ontario or even other provinces. And we want to know, where are they coming from, and how do we alter our management approaches based on these cryptic species or even resistance biotypes? With this, I want to thank you for this time and for this opportunity to present some of the basic results of the research. Thanks so much, and I'll hand it off to you folks. You have any questions?
I think we'll take questions at the end.
Sounds good.
We'll move on to Ashley. So
Alrighty.
Rose, if you can turn off your video, if that's okay.
Alrighty, so switching gears into the biocontrol side of Parvi, I'm going to talk to you about the underground of Parvi control. Did that work? Okay, great. So with any pest, obviously, your chances of biocontrol succeeding or any IPM program succeeding, will be highest if you attack the pest from all sides, at every point of their life cycle. To date, most of the research that we have heard about or found in the literature has focused a lot on the foliage portion of the life cycle, which makes sense. The Parvi do spend most of their life cycle in the foliage. However, they do, similarly to Western flower thrips and onion thrips, pupate in the substrate. So that is another opportunity for us to target them down there. So you would target them in the substrate with things like Stratiolaelaps and Geolaelaps mites, Delocia and nematodes. And then biopesticides are unique in that we can use them both in the foliage and the substrate to target all stages of this pest. So what do we know from the literature so far? There's actually very little work that has been done below ground. Research on soil predators is particularly lacking. We have a few studies that have come out. University of Florida did compare a native Florida species to Stratiolaelaps. And they did find that Stratiolaelaps did eat a fair number of Parvi pupae, But it's unclear how representative this data is because this was laboratory studies done in petri dishes without any substrate present. But Wageningen did release some preliminary data when they were looking at Straiolaelaps and Delocia in substrate lab trials. And they did find that they ate Parvi, but then when they scaled that up to a greenhouse trial, they didn't find any effect. So we also did our own research at Vineland, similar to what Wageningen did in their studies, is we had a substrate cup. So we let the pupate naturally in the soil get down to the depth that they would normally do. And then we applied our soil predators. And in our trial, we didn't find that any of the predators that we tested reduced thrips. So Delocia, Coriaria, Geolaelaps gillespie, and Straiolaelaps immitis, none of them reduced the number of adult thrips that emerged successfully from the soil compared to the control. Now, as we have seen many a times before, what happens in the lab doesn't always happen in the greenhouse. So I think that this does need a little bit of further testing before we can totally rule them out. But so far, this doesn't look terribly promising, but I'm not super surprised by that. We've done work in the past looking at soil predators versus onion thrips. And in those trials, these were greenhouse trials. And we found that Delocia and Straiolaelaps didn't reduce onion thrips populations compared to the control, except in our second trial, we did we had a treatment where we had released Straiolaelaps weekly, and that did show some reductions, but this sort of like apply the predators once at the start, which is a more typical recommended application method, and that didn't have an effect. But in the first trial, we also looked at a drench of Botanigard WP, and the second trial, we looked at a drench of Lalgard M52, and both of those microbial drench treatments worked better than the soil predators, as well as better than the nematodes. So there might be some more promise there. So we did some lab trials to look at the biopesticide drenches against parvispinus. And I'll note that only Lalgard M52 actually has drenching on the label. So in this preliminary study, it was set up the same way that we set up our soil predator trial with the little cup substrate. And as you can see, the Botanigard ES and the Lalgard M52 did seem to work better, but only the Lalgard M52 was statistically significantly different than the untreated control. So one of the possible reasons for this is that we actually used a low drench rate. So we were assuming that growers would want to sprench. So that's why we, instead of the M52 drench rate on the label, which is 10 mils per liter, we used a 4 mils per liter, which is the low end of the foliar spray rate, because we didn't, sprenching at a 10 mil per liter would very likely cause phytotoxicity. So we tested at this lower rate. But we did still see some efficacy of M52, which is great. And then one of the other things that we wanted to look at was how long does the microbial drenches actually last in the soil when you drench them? Because, doing weekly sprenches might be a bit much, both for the plants and just for the growers in terms of labor and cost. So we set up a trial in our greenhouses from February to April of this year. And we had our Mandevilla stock plants, and they're in 1-gallon pots, standard soil as substrate. And we applied everything based on the Lalgard M52 drench rate of 10 mils per liter. And then the other products that we tested, we applied at the similar spore density. And then we collected samples at 2, 4, 8, and 12 weeks after application to find out how much of it was still present and alive in the soil. And so what we found was that all of the products that we applied, so Botanigard ES, Bioseries WP, Principal WP, Mycotal WP, and M52 EC, OD, I should say. These were all still present at 12 weeks after application. So even the products that you saw really tapered down towards, after the 4-week mark, even though those numbers are like very close to the bottom of that graph, they are still present, just at low numbers. But as you can see, Lalgard M52 really stayed consistently high throughout the trial, which is great. It even increased. So Lalgard M52 and Principal WP both actually increased from the 0 week to the 2 week mark, which is cool. And for some reason, I don't know why, Mycotel seemed to like rally at the 12-week point and increase in number. So it's really interesting. But they're present in the soil. They were viable spores, but how effective are they? So we also did a couple of virulence tests using Galeria moth larvae. And so here is a comparison of the number of Galeria moth larvae that were killed by infection at 8 weeks and 12 weeks after our drench applications. So what we do is we take a little sample of the soil, we put them in petri dishes in the lab, and then we add the Galeria moths and see if they die by these infections. And as you can see from the pictures here, the top one is infected with M52. The bottom one is a Bavaria bassiana infection. And so we can actually tell when they have just died by natural causes versus being killed by infection with these microbials. And all of the products at both 8 and 12 weeks did cause mortality in the Galeria moths, so they were still doing something. But as you can see, like the M52 at 12 weeks after we drenched was like killed almost all of the Galeria moth larvae, which is really incredible. And the other interesting thing is that we would expect that this rate of virulence or efficacy to correspond to the number of spores that we found in the soil, but that just wasn't the case. So at this 12-week mark, Mycotel had quite a lot more in the substrate compared to Principal, but at 12 weeks, Principal was clearly killing more Galeria moths than Mycotel was. But overall, across the board, this is really promising for the long-term persistence of these microbial products. And so what does this all mean? So far, our take-home messages are that like the data we have for soil predators is pretty limited, but the earlier signs indicate that they might not be exactly the most effective for Parvi. But we need to do some more greenhouse trials to really confirm that. But Lalgard M52, it both did effectively reduce Parvi and it had a really long persistence in the substrate. Now, these were under optimal conditions. Remember, we did those trials from February to April, so it was like perfect microbial loving temperatures. So we want to do some persistence trials in the summer, particularly in propagation where things are really, really hot, to see if the same persistence works over time. And we also want to look at nematodes. We haven't done any nematode trials yet at Vineland, so that is also on the docket for the fall. And we also are planning to do a greenhouse cage trial, which will compare the efficacy of these various, the most promising substrate-based control strategies to see how they work in a more realistic scenario. And with that, I think I will be passing it on over to Taro to talk about things happening above ground.
Thank you. Yes, thanks everyone for joining us at lunchtime. I will be talking about a trial that tested a combination of mycoinsecticides and predators. So 3 mycoinsecticides and 2 predators were selected based on the results from previous trials. Due to time constraints, I will omit, going over each of them, why they are so good and whatnot. But I still like to point out that among the 3 mycoinsecticides selected, M52 and Botanical DS are the oil-based formulation, and the Principal is powder formulation. And the trial setup was done using the potted mandibula in 6-inch pots, and they were 8 weeks old at the beginning of the trial. There were 4 pots per dome-shaped cage, and there were 9 cages per treatment. And the fertigation was applied automatically via irrigation drippers. And there were a total of 10 treatments, so 2 control treatments. One was untreated control where water was sprayed, and another was a positive control where pylon was used. And for each predator species, Anistis and Aureus, they were either used by themselves or combined with each of the mycoinsecticides. And the insecticides rates were based on Sarah's experience and observation working with commercial growers, except the principle, which is not available in Canada yet. So I just follow the label rate. So the bottom is the trial timeline. So 1 week prior to the start, we collected the thrips and released. So 12 female and 3 males per cage. And at the start of the trial, we did the first application of mycoinsecticides and pylon. And 1 day after that, we did the first release of predators. So the predator release rate was 2 females per cage. Realistic rate would have been 1, but we didn't want that only 1 to die quickly and didn't do nothing for the rest of the week. So we released the 2, just the insurance. And then 1 week later, we did the second application of mycoinsecticides and pylon. And again, a day after, we released the second time the predators. It's the same rate, 2 per cage. And then first sampling happened 2 weeks after the first application of treatments. And remember, there were 4 pots per cage, and we took 1 pot per cage and washed the plants and counted what's on the plant. And then that was repeated every 2 weeks after. So week 4, another plant was sampled. Week 6, another one. And week 8, last one was sampled. So what did we find? So this data is still raw data, and I will present cleaned up version later in the later slides. So don't worry about the details yet. But what I need you to pay attention is the top line, untreated control, the orange line. And as you can see, between week 4 and 6, it declined sharply. And that also happened in some other treatments, although the decline was not as sharp as it was in untreated control. And that looked like it's due to because Parvi destroyed all the fresh leaves and the growth tips of the plants. And that also happened in other treatments. And you can see the extent of the feeding damage. So we counted number of fresh, actively growing growth tips or leaders, as you want to call it, per plant. And then as you can see, over time, except in the pylon treatment, the brown bar on the right-hand side, where the leader numbers are increasing over time nicely. In every other treatments, the growth tips are basically decimated by week 6. And in the untreated control, the light blue bar on the left-hand side, we can see in the week 2. You don't see it in week 4 anymore because Parvi just destroyed all the leaders by week 4. We also tracked the predator numbers. And at week 2, we found more Aureus than the Anistis. So the top 4 lines are the Aureus, and top bottom lines are at week 2, the Anistis. And you can see the Aureus are sitting above the Anistis lines. But Aureus disappeared. I couldn't find any more Aureus at week 4 and on. However, Anistis persisted. And when we excluded the 6-week data because that all the growth tips were gone and the thrips number data was not reliable by the week 6 in most of the treatments, we excluded week 6 data except the pylon because pylon treatment kept the plant quality decent to up to the end of the trial. And you can see it at the week 2 and 4, comparing to the untreated control, the orange bar on the left-hand side. All the treatments tested were significantly less Parvi. However, we saw in earlier slide with the feeding damage figure, none of those biotreatments were able to protect the growth steps or the leaders by week 4. And for some reason, so the dotted line was supposed to be up in with the aligned with the week 0, so that's wrong. I don't know what happened. But basically, if you look at what happened over time, these three biotreatments held their fort or even reduced the numbers of Parvi comparing to the beginning at week 2, although they lost their grip by week 4. Pylon, of course, did good job up to week 6, where the Thrips number started to crawl back. And by week 8, even in pylon treatment, Thrips number ended up more than double. So in summary, we found the Anistis plus M52, Aureus plus M52, Aureus plus Botanical ES. So these, remember, these microinsecticides are all oil-based formulation. And Pylon, of course, successfully reduced Thrips population. However, the biotreatments had only 1 to 2 weeks of residual efficacy, as we saw in the figure earlier, comparing to the 4 to 6 weeks of residual efficacy provided by Pylon. However, efficacy of Pylon weakened by week 6 as the Thrips population began to grow back, indicating the possibility of Parvi becoming resistant to the chemical over time. And because Pylon is limited to 3 applications per crop cycle to prevent pest resistance, the predators and then the predators and microinsecticides can be applied as needed. The results at week 2 indicated that the combination treatment of either predators and the oil-based microinsecticides, M52 or Botanical DS, were as effective as Pylon. So this trial applied treatments only 2 consecutive weeks. So the data suggests that it might be possible to control Parvi with weekly application of those effective biocombinations. And that's it for me. Thank you.
Okay, hi everybody. I guess I'll just keep going here. Hopefully everyone can hear me well, but if not, please someone jump in and let me know. So I am just going to jump in with a few comments related to the situation in our greenhouse vegetable crops in Ontario. Excuse me. In particular, I'm focused on peppers because that is the only crop where I've actually seen Parvi in Ontario. And they did have a common name that was used and probably still is to a certain extent of pepper thrips. So we know that they have the potential to cause a lot of damage in our pepper crops. And I think a lot of our local pepper growers are aware of this pest and, concerned about invasions. We just don't have a lot of experience with it. So I'm, one of the veg people in the group, but I have to say that I'm really grateful for the opportunity to work with all of our flower friends who have the experience and have been working with the flower growers in Ontario to figure out this pest. So we're going to look at a little bit, just briefly, sort of what IPM in peppers looks like and how we can relate it back to Parvi management. And we are begging, borrowing, and stealing from our flower friends throughout this talk. So to start off with, I guess I think it's important to mention that I have only ever seen Parvi in a greenhouse pepper crop once in 2022. And I think it was a really interesting case because the pepper growers sort of set their own thresholds for damage when it comes to thrips. So a common number that I hear is, we can tolerate thrips up to about 10 thrips per flower is sort of, the upper end of the threshold. Some growers prefer lower numbers than that. But what happened was this farm in particular noticed what they said was the same level of damage that they would expect from 10 thrips per flower, but they were only counting 4 in a flower. So that sort of triggered them to look more closely at the thrip species, and they ended up bringing in a few people, including me. Towards the end of that, it had been identified as Thrips parvispinus. So, we do have this question, though, that remains. Were their counts of 4 thrips per flower actually accurate counts? So for those of you that have joined previous webinars, you can see that the way you sample and the way you measure thrips on a plant compared to thrips on a card really is different sometimes for different species or different crops. And so that's something we don't really have a lot of experience with in the pepper crops. We count on cards, we count in flowers, and potentially through the canopy, but mostly those are the two things. So we're not really sure if when we are doing visual inspections of flowers, is that an accurate representation of the numbers of parvispinus. So just, tapping pepper plants may not be that easy. We can aspirate from peppers, but I think we still have a bit to learn as far as how to do accurate counts on a greenhouse pepper plant. But we do use a lot of trapping tools and monitoring tools that are sticky. So yellow cards, blue cards, yellow tape, blue tape. But one thing that has really emerged in our pepper crops over the last decade are echinothrips. And so though, our trapping and monitoring of different thrip species has changed a bit, and a lot of farms are including green cards now, sometimes lower in the canopy. But I think we really sort of need to rethink, the identification and the monitoring and how we're doing it when we very likely have more than one thrip species present, at least, at some point throughout the season. And then as far as bios goes, this is really our primary strategy for managing thrips and peppers. So the minute peppers are transplanted, we usually will see some sort of bio program implemented, and it's not usually, relying on one predator. There are usually multiple predators that are applied to a crop. So I have a photo in there of Rose and I and a former student of mine. We visited this pepper farm that was doing a pretty large scale trial. They split their farm into 2 20-acre sections and went very heavy with biocontrol on in one section. So they had everything from, predatory mites, like they had Swirsky and degenerans and anistis, and they had aureus, and they implemented a Dicyphus banker program, which consisted of mullein banker plants to help Dicyphus establish, and alyssum plants, which is supposed to support, aureus and parasitoids of aphids and things like that. So it was a very large, intense bio program, and it was pretty remarkable to see the how successful it was in that crop. That was pretty exciting. Um, so, thankfully, some of what we've learned from this research is that the biocontrol agents will help. And so a lot of the ones that we're using should help suppress population of thrips, including parvi in our peppers. We do have the great fortune of having biocontrol agents are very easy to establish in pepper crops. It's very different than, say, our tomato plants, which are not really ideal for a lot of our predatory mites. But peppers, we have a lot of success with biocontrol. So again, we don't exactly have a specific program for Parvi and peppers, but I think we can borrow some of the knowledge from flowers and really rely on this standing army approach of biocontrol. And one, I think, sort of word of caution would be also based on some of the research that's been done in flowers that looked at alyssum is Parvi seems to really like alyssum. So I think when we're using it in a pepper crop, I understand the benefits to our biocontrol agents like aureus and parasitoids, but I think we also need to really pay attention to what is establishing on those alyssum plants. So they are thrips magnets, and we do need to really look at the species that are on the alyssum more closely as well. And then the last thing I'm just going to touch on is pesticides, obviously, because these are a part of pepper production as well. So this slide, there's a link at the bottom to an article that was written by Jason DeVoe, who's a colleague. He's the application technology specialist at OMAFA. So we did a really fun spray day last year at a pepper farm, and he got to play around with their equipment, and he made a lot of tweaks, and it really improved the coverage that they were getting on their crop. So I encourage everyone to read that article because it really there's a lot of good tips, I think, and things to look at. But coverage really matters, and this is where it's so important to understand the biology of the thrips and where they're hanging out. So we have, our western flower thrips, maybe eastern flower thrips is there now too, that are hanging out in the flowers. We have echinothrips that is all through the canopy and really does actually like the lower canopy in a lot of cases. But so coverage sometimes really matters, and that, we tend to focus a lot of our scanning at the top of the plant. These are really tall plants. We have an 11-month long crop cycle. And so, we probably need to pay more attention to what's happening throughout the canopy too. And we've learned that from our situation with echinothrips. And that's a video of our spray, uh, automated spray machine. So the last thing, this is another thing I borrowed from one of Sarah's blog posts, the rotation guide for Thrips parvispinus. So what I did, I just looked at all of the products that were in that Thrips rotation. And if there's any veg growers on here and you want to look at those details, there's the link below. But I looked at the products that were on the Thrips rotation, and I compared it with the pepper, what's labeled for peppers. So I think the good news is that we do have a lot of products that could support thrips management. So if Parvi is detected in a pepper crop, we know that we probably need to pay close attention to how we're measuring them on the plants. We also have biocontrol agents that we can use to support the management and then incorporate based on some of the other research that you heard about today too. Start off with some microbials and rotate in the stronger chemicals as needed. So that is all I have.
Great, Cara. Sorry, I'm just gonna figure out how to turn my camera back on. I feel like I had trouble with this last time too. Oh, thanks, Marky. That was helpful. Okay. So I need about 15 minutes to wrap this up. I'm going to talk about how we put everything together that Ashley and Taro talked about in terms of an IPM program for Mandevilla. Uh, just if you need to leave at one, no worries. This is being recorded and it will be posted soon. But we hope you'll stick around and join us for questions at the end. So, just summarizing what we found for the floriculture industry, Ashley's work showed that a lot of the predatory arthropods may not be so good for Parvi, although we should test that more in commercial greenhouses. But some of the tests I've done before, they weren't really recoverable and didn't seem to be doing much. But it really sounds like microbial drenches, mostly M52, but some of the WP formulations gave good bang for the buck. So I do recommend putting that in the program. And then on the foliage, big bios plus microbial seem to be really promising. But as Taro mentioned, it has to be the right combo. So here, the wettable powders didn't seem to be doing much. So it was really the oil formulations plus Anistis aureus. So what does this mean for IPM in Mandevilla and other tropical plants? One of the biggest factors I think we need to think about is that we can't just cut and paste bio programs like we do for Western flower thrips into other crops. It's not just like a mite sachet plus sticky cards or something like that that works for a lot of things. We're also going to have to get into the mindset that we're going to need to do weekly releases of natural enemies, as Taro mentioned, to make sure that control stays, as opposed to, for Western flower thrips, we can, put a mite sachet out. We're probably also going to need to add in compatible pesticides such as Rimon and Ferrants to support these bios in their job. And I'll talk a bit more about this with some examples from on-farm trials. And we're also going to need to make use of other tools in the IPM toolbox again to support these bios and make sure we're getting our money's worth out of them. So some examples of on-farm trials that have been done in Ontario. I've talked about this before, but this is Judy Cawley's work from Plant Products, where she was able to successfully control Parvi and anthuriums using aureus. And initially, she tried really high rates of aureus, and that did work, but it wasn't really economically sustainable in the long term. So then what she found was that if when the plants came in, if they were first treated with success and then 3 applications of M52 and then followed with a lower rate of aureus, that actually was quite effective. So again, an example of using softer chemicals to sort of knock the thrips back and then using your bios. And in terms of using another tool in the IPM toolbox, cultural controls were really important here. They had a variety, a purple anthurium that seemed really susceptible to Parvi damage. And even at the high aureus rates, they just weren't getting good enough control of that variety. So once they decided not to grow that variety anymore, the program was really successful. Here's an example of a trial I did in 12-inch Mandevilla in 2023. So this was before we had some of this good information about the bios, which were sort of better than others. So this is really a spaghetti at the wall trial where we did dip our cuttings in oil, though, not in sort of like the best combinations that were talked about in the first webinar in this series. And we did economical rates of bios. These were donated by BioBee. So we did things like a theta, hypoaspis, and aureus. So now we know that aureus was probably doing the heavy lifting there. And we did add in weekly microbial sprays, but this was of a Bovaria wettable powder. And we were using yellow sticky cards, which again, we found since then that blue is a little bit better for Parvi. So you can see that this program, even though none of the components were ideal, did suppress Parvi for like a little bit. But then once temperatures really took off in August, we got up to 85 thrips per plant. I also went to France. So other lesson when you're in the middle of a trial, do not go away. But what's really important here is you can see these numbers came back down after we did some treatments in September and October. And we did this almost completely biologically. So we used sprenches or drenches of M52 3 times at a 1 ml per liter rate throughout all of these 12-inch. We also added Anistis at 1 per square foot for several weeks. And then we added in blue cards for mass trapping. And you can see that all of the Parvi went down significantly. and pretty much went down to zero without help in the other colors, except for the white, Summer Star White. You guys have heard me talk about this one before. It seems to be able to support much higher numbers of Parvi for reasons we talked about in the second webinar. So that particular color needed a spray of Remon, which is another softer IGR chemical, to really bring down the levels. But using all of these in combination, we were able to get the thrips numbers right down. So the fact that we were able to rescue this kind of like, not great situation that I keep using as an example, with these microbials and bios means that if we had used them from the beginning, along with a better dip solution, we probably would have gotten really good control and kept at low levels throughout that period of the crop. So knowing what I know now, also working along with closely with a couple of Mandevilla growers for the past few years, I've sort of combined what we're doing now in production with some of these new pieces of information and how I would fit them together into an IPM program. So we need to think of our long Mandevilla crop as having distinct biocontrol windows where we have opportunities to employ these biocontrol agents. So the first one is obviously cutting dips and then in propagation. So absolutely we should be doing dips, and Ashley found that the combo of Suffoil and Botanigard did really well and had low phytotoxicity. So that can help us start clean-ish. That's not going to completely eliminate things. So what about other bios in prop? So things like Aureus anistis, microbials, even nematodes. Unfortunately, we've tried some of this. Again, with BioBee, I did a trial where we really released some big bios in propagation in Mandevilla. But the problem is that because of the time of year and because of how Mandevilla like to be grown, usually intense, propagation can reach temperatures at 40 degrees Celsius or above. And if you look at this handout made by Michigan State Extension and OMAFA and Kansas State University, we've put in the optimal temperatures and the max temperatures for the commercially available bios. And you can see that both arthropod and microbial natural enemies really don't function much past 30. Some of them make it up to like 32. So when we did the trial with BioBee, we never recovered any of the large bios from propagation. So our next step is to validate whether microbials and nematodes, which are applied to the soil, might be a little bit more protected, and some of them might still live, but that's upcoming. So for that case, I would say for now, this coming season, I guess that we're in right now, I wouldn't be adding anything to propagation just yet. So that means our most important biocontrol window is our second biocontrol window, which is early production, which by that I mean just after potting up, but before spacing of your Mandevilla pots. And obviously we want to get on top of Parvi early, but honestly, one of the biggest drivers of doing biocontrol here is going to be two-spotted spider mite and to a lesser degree oleander aphid. And that's because we have been able to successfully control Parvi for a couple of years with just chemical insecticides, but the two-spotted spider mite gave us a break for a few years while we figured it out, which is very polite of them. But now they seem to be back with a vengeance, and they seem to be coming in resistant, possibly because of sprays happening on mother stock before we even get the cuttings. And then as soon as we do a chemical spray for Parvi, they're just, ready to go. So the two-spotted spider mite opportunity to do biocontrol is really going to be the driver of also doing Parvi biocontrol, in my opinion. So what would that look like? So for Parvi, again, sort of like Judy Colley's work in Anthurium showed, we want to knock back the Parvi first with chemical applications. So what I found to be really effective at some of the farms I've been working with is a sprench of Contos at the end of propagation. And this protects against both Parvi and oleander aphid. And then once the cuttings have been potted up, then you want to follow up at potting, but before spacing, with either another application of Contos via spray or 2 applications of Remon. And all of these are attacking the Parvi larvae, which is the stage that's likely to be highest after they emerge from eggs that are present on the cuttings, because the dips will take care of most of the adults and the larvae. So then in terms of biocontrol, if you wait 2 weeks after the Contos drench, you can add Aureus or Anistis. And I would also encourage you to do sprenches or drenches of M52 at that 4 ml per liter rate, which is probably more economical than doing the full drench rate. And that'll stay in the soil for quite a while, as Ashley mentioned. And potentially that's where we could add in the nematode Carpocapsae, which studies in Florida showed were one of the most effective species for Parvi. In terms of spider mite biocontrol, here you would want to add loose folaces. And the reason we would pick folaces is it's a bit more tolerant of pesticide residues than Persimilis is. And we've also found that M52, which has spider mites on the label, is a really effective product for spider mites. You just have to use it at a bit higher rate, so more in the 2 or 3 mils per liter. So you may want to do bi-weekly applications of that for spider mite suppression, but it'll also be suppressing your Parvi. So a lot of these chemicals that we have are sort of like you get, hit 2 birds with 1 stone sort of mentality. And also, I just want to make a note for Laugard M52. It does come in a smaller bottle size now, a 250 ml bottle, which is good if you're doing smaller areas like prop or plants before spacing, because after you open it, it's got a really short shelf life, probably like 3 days in the fridge, and then you really have to use it up. So if you're not able to use that big bottle up, please consider buying the smaller bottle. So now we're in our chemical window. So unfortunately, we're going to be spraying and knocking back any of our bios that we were introducing weekly, like Phalasis and Aureus and Anistis, because we really need to do a cleanup before Mandevilla enter the cold growing period. So in December and January, they get grown anywhere between 14 and 18 degrees Celsius to put on slow growth. And this temperature is just not amenable to any of our biocontrol agents, not even really microbials. We need at least temperatures of 20, 22 to make them effective. And you can see here what Tara was talking about the leaders sometimes just don't grow or get damaged. You can see the damaged leaders in these photos. So if you don't do that pesticide cleanup in the fall, the leaders just won't grow or they'll be too damaged because even just like 1 or 2 larvae on those leaders can destroy them. So what we're doing is pretty standard in the industry now is 2 pylon applications at a 1 mil rate, so the same as Taro used. We're doing 2 sprays 5 to 7 days apart. And then the other thing we're doing is adding in a mechanical control, which is to remove the flowers of the Mandevilla, and that's for 2 reasons. Those trumpet flowers, man, it's really hard to get contact insecticides into those. And even though pylon is translaminar, pesticides just don't translocate into petal tissue the same way they do onto leaves. So removing those flowers and bagging them up really is almost like a 3rd spray. So just a note about spray coverage and thrips. So the reason I'm recommending 2 sprays of pylon 5 to 7 days apart is because the first spray you're going to do is going to hit any adults and larvae already on the leaves. And if we just did this, it would last about 6 weeks. But remember, as Ashley pointed out, thrips have hidden life stages, including Parvi. So the second spray will hit any adults that are hatching out from pupae, which have been hiding in the soil, as well as newly emerged larvae that are hatching out from eggs embedded in the plant tissue. So this is how we get the product to last 12 to 14 weeks. And the difference between the cage trials and the greenhouse trials just might come down to equipment or, spray coverage, being able to go up and down both sides of the rows, or just, the differences between cage trials and greenhouse trials. That's just how it goes sometimes. And you may be thinking, oh, like 2 sprays of pylon back to back, that seems like a lot. Well, this technique actually reduces the number of sprays you needed for control compared to a weekly rotation. I have one grower that I've been working with, and they went from basically spraying every week for the 16-week period they were growing rooted liners, and now they're down to 6 sprays total. So doing this back-to-back application of one chemical before you move to the next is highly effective, and it doesn't promote resistance as long as you do the 2 applications within one pest window, which is the generation time of the pest. So for Parvi, this will be 12 to 14 days. So if we did a 3rd application of pylon after the 14 days, that could potentially be promoting resistance. So you want to switch to a different AI. So our last biocontrol window is late production. So basically in February, as soon as the sun comes out and things warm up in the Netherlands and here, we've noticed that's when Parvi likes to sort of relaunch itself, like a little Parvi 2.0 infestation. So what you can do here is, again, if you want to go a little bit more of a chemical route, you can knock back the Parvi larvae, because again, that's the stage that is going to be the highest right now, with Rimon or a spray of Contos, as long as you're not exceeding your maximum applications for the year. And the Contos will take care of both the Parvi and any aphids that might be around. And then we also recommend during this period doing biweekly sprays of M52 for both two-spotted spider mite and Parvi. And just a note though, at this later crop stage, you want to do 3 applications of this max in a row. And then you might start to see a little bit of phyto because of the oil buildup. So you either want to take a break or maybe spread these sprays out a little bit farther. And then for two-spotted spider mite, this would be the time to turn to Californicus sachets in the large size pot. Unfortunately, Phalaensis doesn't come in a sachet, but you can broadcast Prisimilis depending on the chemical applications you've done for the smaller pots to help save money. And just credit to Sarah from Global for helping me with some of the spider mite options out there. And hopefully, if you've done this whole program, you won't really need to clean up your crop for either Parvi or spider mite. But if you do, you've done so few chemical applications. We really only relied on 3 chemicals for Parvi. We've only been doing biocontrol for spider mites. So your cleanup pesticide should work. And the last thing I'm just going to mention is I really still feel like Parvi IPM is much like the Swiss cheese method we thought about for COVID, where even if you've got strong pesticides or like vaccines against COVID, it was, to really make sure you didn't get an infection, you needed to make sure you were washing your hands, wearing masks, being in a ventilated space. So we're still sort of at that place with Parvi IPM. We don't have a really solid biocontrol program yet. So we really need to make use of all these different tools from the beginning of cutting dips to mechanical controls like mass trapping, to making sure we're picking the right bios and combining them with the right pesticides at the right time. And that will give us a high quality sellable crop. And with that, I will end the presentation just thanking our research project that was funded by the Agri-Science program, an initiative under the SCAP program, and we'll open it up to any questions.
All righty, thanks, Sarah. So as she mentioned, it's time for questions. If you have any to enter into the Q&A, we can answer them live. And if you would rather, for instance, if you're on a phone, you can put up your hand, we can unmute you and ask live. Nothing yet. And as I believe Sarah might have mentioned this, we mentioned it in previous presentations, I'm also happy to put people in touch with the researchers directly if you want to ask something offline. So all you need to do is respond to the invitation that you received in your email, or you can email admin@fco.ca, and I am happy to pass those questions along to the researchers.
Yeah, and in terms of the recordings, you can find them on the Focus on Floriculture YouTube page, which is a Flowers Canada sponsored YouTube page. And then also on my On Floriculture blog, there's a Parvispinus tab. And I will embed the videos there, and all our previous webinars and links to other people's outside webinars are also linked there for Parvi. So I just wanted to keep all the Parvi information in one space.
That's excellent. Thanks for doing that, Sarah. And we'll also be sending those out to our members by email. So all of the resources will be sent to you via email, as well as an exit survey for the webinar. So watch for that. There'll be one at the end of this webinar, but there'll also be one emailed out. So one or the other, not both, please. All right, looks like we don't actually have any questions. I don't know if there's any questions amongst the panelists. Doesn't seem like it. Well, perhaps we will wrap it up. I want to say thank you to everybody for joining us today. Hopefully you've learned a little bit more about the potential for IPM solutions for Thrips parvispinus. And if you have any further questions, as I said, please send them along. You may think of something later. Thanks to the team at Vineland, OMAFA, and AAFC for making themselves available to speak to our growers today. All of these partners are big supporters of the industry, not only like by doing events like this, but also through sponsorship of FCG, which helps all the growers keep growing and expanding. As always, FCG is here to support our members, so don't hesitate to reach out. And I hope you will join us for our next webinar. Have a great day.